Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Calcium Signaling in Neurons and Glia in the Mouse Brain During Exploratory Behavior. I'm Andrew Davies from the events team here at scientist.com, and I'll be your host today. This webinar has been sponsored by Neurotar, so a big thank you to them for helping make this event possible. We're very fortunate to be joined by Dr. Alexey Semyanov, Professor and Director of the Neuroscience Center at Jiaxing University, and Dr. Amit Agarwal, the Chica and Heinz Schaller Research Group leader at the Institute for Anatomy and Cell Biology at Heidelberg University. They will be discussing their research on activity and calcium signaling during locomotion in the brain of awake and head-fixed mice. Just before we get started, I'd like to cover a few brief housekeeping notes to help you get the most out of your experience. First, this webinar is being recorded and will be available to view on demand shortly after the live event. Next, if you take a look at the interactivity panel on the right-hand side of your screen, there are 4 tabs corresponding to chat, Q&A, polls, and handouts. If you experience any technical issues, please first try refreshing your browser, as this fixes most problems, but if you're still having trouble, please reach out using the private chat function and we'll help you to troubleshoot. We'd also love to hear from you, so please send any questions, thoughts, and comments using the Q&A tab, and we'll address as many of them as possible in a Q&A session following the presentations. And finally, you can explore relevant resources in the Handouts tab to learn more about some of the content presented today. But just before I bring on our first speaker, we have 2 brief audience polls for you. Our first question is, which of the following research methods do you currently use? And please select all that apply. The options are 2-photon imaging, electrophysiological recordings, behavioral assays, optogenetics, functional ultrasound imaging, or wide field imaging. And I'll just give everyone a moment to submit their responses. And once again, please select all that apply. And thank you very much. And our second audience poll is, which statement best describes your current situation? And here the options are, I currently work with awake rodents and am interested in head fixation techniques. I will work with awake mice in the future using optical or functional ultrasound imaging. I will work with awake mice in the future for electrophysiological studies. Or I just have a general interest in the subject and technique. And I'll just give everyone another moment to select their response. And thank you all once again. Without further ado, it is my pleasure to welcome our first speaker, Alexey. Alexey, take it away whenever you're ready.
Hello, everyone. It's my pleasure to be here at this webinar, and knowing that so many people decided to participate in our webinar. I will be talking about calcium responses in astrocytes and neurons. But before going to our experimental data, I would like to start with some introduction and put all our studies in a general context and describe the framework of our research. And I will start with this introductory slide showing where all this started. It all started from description of brain cells, and we should thank this Spanish scientist Santiago Ramon y Cajal, who used modern optics to his time and staining methods suggested by Camillo Golgi, Italian scientist. And Santiago Ramon y Cajal actually described and drew by hand most of the cell types in the brain which we currently know. He described neurons, glial cells, cells of blood vessels, and since his times, we started to look more into function of these cells. And it happened that one cell type was very special. These are neurons. Neurons are electrogenic cells. So these cells are able to generate action potentials. And neurons connected to each other by plastic synapses. And these 2 properties of neurons made them special in terms of people start to think that maybe this is the basis of the brain function, and all function of the brain can be explained by neuronal interaction and function of neural network. However, a few decades already, we are changing this focus and start to concentrate on different interactions in the brain, not only within neuronal network, but also at neuroglial interactions. And this is the schematics showing very well-known phenomenon or very well-known concept of tripartite synapse. And this tripartite synapse evolved, and now people start to talk about multipartite synapse. And here you can see the general idea of this. So presynaptic neuron, postsynaptic neuron, and perisynaptic astrocytic process, which is called leaflet in this particular case. And during synaptic transmission, neurotransmitter not only targeting postsynaptic receptors, but also receptors on the leaflet, on astrocytic process, also taken by transporters to astrocyte, and this all causes calcium elevation in this leaflet, and this in turn triggers release of glia transmitter, which regulates synaptic transmission. So this is integral part of the synapse. If you go to the level up, so we can talk about regulation of multiple synapses. For example, calcium can cover larger territories of astrocytes, which would modulate signaling within synapses, which might belong even to different neurons, which are not connected synaptically to each other. And then if you go to the level up, so we can talk about interaction between neuronal network and network of astrocytes. Astrocytes also connected to each other by gap junctions. Calcium activity within astrocytic syncytium astrocytic network also can cover larger territories, which in turn would modulate large areas within neuronal network. However, this is important type of communication in the brain and related to calcium, which we will be talking about today. Astrocytes communicating not only to neurons, astrocytes also communicate to other elements of the brain or other cell types in the brain. So this is a schematic showing different type of interactions of astrocytes. They can interact with blood vessels, with microglia, oligodendrocytes. They can release components of extracellular matrix. They regulate extracellular space by shrinking or by expanding their processes. And astrocytes also respond to all different types of signals. So we already discussed that astrocytes respond to neurotransmitters released within synapses, but astrocytes also respond to neuromodulators, for example, to noradrenaline, serotonin, dopamine. Astrocytes respond to cytokines, hormones, growth factors. All these chemical substances also target neurons and other cells in the brain, but there are astrocyte-specific signals. So astrocytes bring this information to the network. So this partial pressure of oxygen in the brain tissue, it's intracranial pressure, vasomotion. So astrocytes equipped with mechanoreceptors like piezo-1 or TRPV receptors. They also sensitive to temperature. And all this triggers calcium response in these cells. And based on multiple interactions, on different type of interaction within brain, so there are several concepts suggested. Not only multipartite synapse, but also such concepts like neuroglia vascular unit, extrasynaptic signaling, volume transmission. And if you look at this concept, and that's what people study within the particular research projects, so you will see that maybe the same neuron or the same astrocyte or the same type of signaling is involved in each of these concepts. So if you look in isolation, so you probably miss something more important happening within the brain tissue, within this particular structure. And so based on this idea, we suggested a unifying concept, which we call brain active milieu. And that was published in 2021 in Teens, in Transition Neurosciences. And then later next year, in 2022, we had another story published in Function, where we gave more sort of precise description of what is the brain active milieu. And also we had this beautiful cover image already developed by the artist from the editorial board. This is three-dimensional representation of the brain active milieu. And what we say here, that brain is works as a structure built of multiple cell types, neurons, glia, and non-neuronal cells, and non-cellular elements like extracellular space and extracellular matrix. All these elements interconnected in space, and interaction between all these elements is very important and defines the brain function. And so with this in mind, we set our projects in a way that we're not only looking how physiological function or different type of behavior affect neurons. We also look at other cell types, and today I will talk about two cell types, astrocytes and neurons. So, and this is the way how we perform our experiment. This is the scheme of experiment. We use mice and stereotaxically do injection of AAV, adeno-associated virus. This could be all different type of viruses depending on the question. In this particular case, on this schematics, you see injection of AAV5, which triggers expression of GCAM6 calcium sensor under astrocyte-specific promoter, can be on neuron-specific promoter. Then we put on the craniotomy window, we put a glass window and a glue head stage. And then after a few weeks, when animal recovers, and then when expression of this calcium sensor occurs, so we do handling and habituation of animals to the experimental system. And this is the drawing of our experimental system, which is already introduced. This is a Neurotar airlifted platform, and the concept I will describe on the next slide. So this is how the surgery look like. This is stereotaxic system with injection of the AAV. This is during surgery. Animal is anesthetized, isoflurane anesthesia, and then after the surgery, animal recovers and already wakes up with this cranial window and the glass and this head stage holder. And so then animal feel pretty good after the surgery, so they fully recover, and then after a few weeks we start the experiment. So this is experimental system, how it looks like. So this on the left side you can see the whole setup on the anti-vibration table, then 2-photon microscope, and this home cage airlifted platform from Neurotar. And so next, it's a small video showing how it all operates. So I will show you. First, let me explain. So this is like a table or breadboard with many small holes in it, and the pressurized air goes through these holes and lifts up a very light open field platform. So this is carbon made, and so it's very light. So it's floating on the cushion of the air. And animal being head fixed can push this platform by the puffs, and so this platform is moving under the animal, and animal get impression perceive it's like the animal is navigating on this platform. So let me show how it works. But animal is of course head fixed and remain in the same place under the microscope objective. So here's the video. You see a mouse, it's head fixed. And so moving on the platform, but in fact it's not mouse moving on the platform, but platform is moving and mouse thinks it moves. It's a little bit like in a train, like you sit in the train and you see the nearby train is start to move and you don't know whether your train is moving or the neighboring train is moving. So and what we do next, we track the animal position, track the animal movement, speed of the animal, and look at the calcium activity and calcium response in cells, in astrocytes in this particular case. So what you will see here in this circle, there is a color spot, it's a mouse. So then the mouse start moving and you will see the trace. And then at the same time, look at the right side, this will be a movie showing the calcium response in astrocytes. Okay, so now and it's already started. So the animal was stationary, then when it start moving, so we can track the speed, and then we can also see calcium response in astrocytes. There are quite a few astrocytes here on this image. So of course we're not getting that beautiful picture straight away, so I have to process these images, and so this is the image analysis pipeline. So you will see 3 videos here and 1 bar at the very right side, on the right hand side. So the left video, it's raw images. So raw video, how we see this when we record this fluorescence. Then in the middle, it's a denoised video. So you will see more of the structure. And then on the right side, you will see delta F over F. So only change in fluorescence. So this will be showing you the response, calcium elevation. So let me start. So no raw image is very noisy. So then animal is, and this bar actually on the very right side is the speed of animal. So when the bar goes up, animal is running. So when animal is running, you see a very clear calcium response in astrocytes. When animal is not running, so response is very, very dim, so there's not so much of activity going on. So then what we can do, we can represent this video for the picture for in the publishable format, so which you can print. So in this three-dimensional graph, so this is x, y. So this is surface from which we do sampling, and time. So, and you can see where the calcium activity starts and how it changes in time. And this blue bar on the right side shows the animal speed. So when the bar on the right side is blue, the animal is not running, and then you can see the color code. And change the color so the animals start to run. So, and you can see episodes of running of animal, and you can see when the animals start to run, we can see a significant increase in calcium activity within astrocytic network. And you can analyze this image, you can analyze this video frame by frame, and you can detect delta FORF, these changes in fluorescence. Then you can see the active area covered by calcium activity within each frame. And then you can, of course, try to correlate it to the animal movement, to the speed of animal. So there are three different parameters. On the bottom, it's mouse speed. And then in the middle is active area, part of the frame covered by calcium activity. And on the top, it's a delta FORF, a change in fluorescence, the mean delta FORF from each frame. Of course, this is a wonderful system. This is a lifted platform from Neurotar, which allows us not only measure the response to locomotion, but also it allows us to track the animal. So, and what we can do here, we can basically look where the animal is located in each moment in time, and what the speed of animal and how calcium activity changes in this particular point on this platform. But what I didn't tell you when I explained the experimental setting, I didn't tell that we work with somatosensory cortex. So this somatosensory cortex doesn't supposed to respond to the animal location on the platform. So we didn't expect to see any kind of space coding with astrocytes and somatosensory cortex. So we should probably look in hippocampal astrocyte if you want to ask such questions, but you can see this beautiful coding and traces of an animal, and you can see where the calcium activity changes during animal locomotion and how it relates to the speed and position of the animal. And in addition, so we can look not only at astrocytic network, astrocytic in situ, we can look at individual astrocytes. So we zoom in. And so this is one individual astrocyte which we look at. And so we ask a question, where the calcium activity starts during episodes of locomotion. And so here in the middle, you can see astrocytic soma. And then this is astrocytic domain. And we made this wide line scan. So basically this like a rectangle structure, so which way we can sample calcium activity along long axis. And then we can plot it as a line scan. And so 0 distance means middle of the soma, and yellow dotted lines, horizontal dotted lines are the dimensions of soma, sizes from left to right of this line scan. And so then we can look how calcium activity develops within this line scan during episodes of locomotion. So, and underneath you can see animal speed, and so how it changes where the animals start to run when it finishes to run. And so when you look at the calcium response, you can see that it starts first at the distal parts, at the distance from the soma, and then start to propagate towards soma. And in soma, it's become brighter and larger. There is amplification of the response. And so you can see it also if you take it frame by frame. So there are several frames here from 1 to 6. And so you can see first before animal locomotion pretty silent astrocytes, so there's no much of calcium activity going on. When the animals start to run, there are some episodes of activity starting at distal processes. Then they start to go closer to soma, and then eventually they emerge in soma, and you see amplification, a kind of calcium surge. And then animals stop running, and then it's also fades, so it decays the response. So then the process this activity. And so what we've done next, so we identified two regions of interest, soma and the processes, and we wanted to compare calcium activity in these two regions of interest. And so this is normalized response, the amplitude normalized. So what we see here, that response in the process starts earlier than in soma, and we can measure the latency. And this latency is actually very variable between different astrocytes, and usually like a few seconds. So it's a quite slow process. So this calcium activity integration in astrocytic soma is not very fast. And probably animal already, in some cases, even finish running while calcium activity is still being integrated in the cell. So then we looked at the amplification of the response. The response gets larger, approximately maybe 5 times larger. So it's a significant increase in amplitude. And what we also found was quite interesting, that during quite long periods of running, we start to see oscillation of calcium activity in astrocytic soma or astrocytic somata, which doesn't happen in processes. It might be reflected to processes a little bit, but it requires integration. And so usually people see calcium oscillations in cultured astrocytes. When people look at the calcium activity in vivo, there is general belief that this calcium activity is rather stochastic or related to certain activity of neuronal network, release of neuromodulators like noradrenaline, etc. But there is still ability of astrocyte to generate these oscillations, and this can be recorded in soma during prolonged episodes of calcium activity. Okay, so I will not stop any longer here because we're limited in time, and I want to move to another cell type. I want to talk about neurons now. So this is pretty much the same idea, the same experimental setting, but we use different virus. So we express calcium sensory neurons, but it's the same depth in the somatosensory cortical layer 1. And so here we don't see somata of neurons. So we see somata of astrocytes, but we see only neuronal dendrites. And you can see many of those dendrites in this region. And let me start the movie. And so you see animal again on within the circle. There is a spot showing the animal, and then we can look at the calcium activity in neurons. So, okay, you see animal is stationary, but there is a lot of activity in neuronal networks, in neurons here. So, and when animal is running, you barely can see increase in calcium activity within this neuronal, with this sort of neuropil, let's say. So there are many different dendrites emerging from different neurons. So okay, so let's look at the reconstruction of this calcium activity in 3D again, XY time design. So you see the same idea as for astrocytes, and this animal run several times during this recording. And what you can see here, there is already striking difference. There is a lot of activity in neurons during quiescent period. And during locomotion, you can see some increase in this calcium activity, but it's not such a striking difference like in astrocytes. And this is the summary of this activity. Again, this animal speed, several episodes of locomotion, active area, and delta F over F. And so what you can see from here, there is a lot of activity in between episodes of locomotion. And so what we ask next, whether this is the case for all different type of structures, what we see in this image, whether all neurons respond the same way. It's like synchronized activity. And to address this question, we used open source library from the program, which is called Cayman. which is abbreviation for calcium imaging data analysis, which was published by Andrea Bevanucci and eLife and colleagues in eLife in 2019. And so what we use it for, this software is good for detecting of individual neurons and getting calcium activity in vivo from the neurons. But we don't see somata here, but this algorithm was quite useful for identifying regions of interest which are synchronously active. Sometimes they are not connected, but this does matter. So maybe these are just different dendrites coming from the same neuron within this focal plane. And so these are several regions of interest, which we think may be belonging to the same neurons. And so then we looked at the calcium activity within this region of interest, and we found that there are indeed different neurons. Some of neurons are active during episodes of locomotion and silent during quiescent period. Some other active during quiescent periods and not during episodes of locomotion, and some other active during both periods of time. So this is interesting phenomenon, but so far we don't have a good explanation for that. Maybe this we can address in the future work. But at this stage, I would like to move to the probably most interesting part of my presentation and show you a comparison between neurons and astrocytes. We did different we compared astrocytes and neurons in several different ways. Firstly, we introduced the ratio, which we call quiescence to locomotion ratio. So we measured delta F over F for active area during quiescent period and during locomotion period, and then we calculated this ratio. Because neurons were active during quiescent period, this quiescent to locomotion ratio was different from astrocytes, which was silent during quiescent period. And this summary data showing that in neurons, quiescence to locomotion ratio was significantly higher than in astrocytes. Then we, and for both for delta F over F and active area, then we look how calcium response in neurons and astrocyte correlates with animal locomotion, with animal running. So we did cross-correlation analysis for calcium response, and we found that neuronal calcium response precisely timed with animal locomotion, while associative calcium response lagging. So it actually starts or actually occurs a bit later. And so this is also showing here correlation coefficient was quite high in both cases, but lag was higher in astrocytes than in neurons. In neurons was no lag. So there was precisely timed response. And another thing which was quite interesting and quite novel, that when we looked at the paired run ratio, so those people who work with electrophysiology, they know paired pulse ratio. So when you stimulate neurons twice, and then you measure the response for each of these pulse. So here, this is recorded in vivo, and we had natural stimulus, run of the animal. And so we look neuronal response for 2 runs, which were closely timed, and astrocytic response in 2 runs. And what we found, that actually neurons responded each time pretty much with the same amplitude of the calcium response, while in astrocytes we had a reduction of the response, which might reflect refractarity of calcium activity in astrocytes. And this is summary data showing delta F over F paired run ratio and active area paired run ratio. In neurons, it's all around 1, so this is the same response on each locomotion episode. In astrocytes, it is significantly diminished. Okay, and this is the summary of my talk today. I already introduced all these points, and I will not going to stop here for a long time. You can stop your video and read if you like. And also, I would like to acknowledge people who are involved in this work, and Alexey Semyanov for data analysis, Anna Fedotova, Maxim Doronin for recording of this calcium activity, Alexey Verkratsky for developing concept of the brain active milieu. And of course, I would like to thank people from Neurotar who helped us to set up all these recordings in their lab first, and so which we continue to use in our lab now. So on this, I would stop, and I would like to transfer and give stage to next speaker, Amit Agarwal. Amit, please.
Perfect. So thank you very much, Alexey, for passing on the baton to me, and a fantastic talk. As you know, that my lab also studies astrocyte. It is always nice to see still a lot of new data coming on astrocyte calcium imaging. But today, I will not be talking about astrocyte, but another type of cells which my lab concentrates on a lot is oligodendrocyte lineage cells. So these are the precursor oligodendrocyte cells, which we work on. And these are really new discoveries, which we will be presenting first time out. And our manuscript is soon out for everyone to read and check out. So in this study, what we were trying to look at is a new concept which is there, which is emerging, is what you call adaptive myelination. So in general, we know that our memories are stored as synapses. And there are long-term memory storage with more stable synapses, and there are transient synapses. But there is another way of storing memories, and this can be stored through putting new myelin sheath. And this is what you call adaptive myelination or adult myelination. So just to give you an introduction, what it means is that, for example, let's say that there are these 3 neurons, which you can see here. And this neuron is firing action potential, and the action potentials can travel at very high speed because of the myelinated segment, and can reach the neuron. But this neuron is not getting enough excitability to propagate the information further on. This neuron is also getting activated, but it is a bit more sluggish because it does not have everything myelinated, and there are still larger gaps, these not nodes, but gaps, and that does not let the action potential to propagate to the next neurons, and this cannot be engaged. But there is a way of making the whole thing fast. And the way of making things fast is that the first question here arises is that whether these gaps exist. And for a very long time, it was thought is that there are no gaps. In the adult brain, these gaps are all filled up and everything is fast and fine. But this is not the case. As it was shown in the study published almost 10 years back in Science using the serial EM sectioning data, that there are myelin segments, and between the myelin segment there are very large gaps which can be filled, which can be there, but the real gaps which lead to the propagation of action potential fast, the so-called nodes, are quite tiny. So these are large gaps there. So that means that there is a place during the still in our adult brain which can be filled up with myelin to make things faster. So it has been shown by several studies over time that when you stimulate neurons, when the neuronal activity increases, there are formation of new myelin. And the question arises is that how this myelin is formed. There are various views over it, that probably the already existing oligodendrocyte can lay down the new myelin sheath, put a new projection and lay down a new myelin sheath, or there are class of cells which are called oligodendrocyte progenitor cells. And these cells can differentiate into oligodendrocyte, forming new oligodendrocyte, and these oligodendrocyte make new myelin sheath. And once the myelin sheath is laid down around here, as you see, the gaps are filled up. The action potential can now reach this neuron in synchrony with the other action potential and engage the neuron and hence transmit the information. And the myelination is quite stable. And with this means of myelination, laying down new myelination circuits, and the activity and connectivity can be led to more permanency. But here are a bigger question. The question is that if OPC needs to change its fate from being an OPC to an oligodendrocyte, it must sense the activity. And what modes of neuronal activity tells an OPC to change its fate is a big question. That the cell has to change its fate, and what kind of activity it senses remains unknown. And the other question is that how OPC integrates this neuronal activity to have these fate changes. So this is what we are trying to answer, some aspect of these questions in this following study. So a quick introduction to OPC. Oligodendrocyte progenitor cells, as the name says, they constitute roughly 5 to 7% of all brain cells in our brain. And they are very beautifully tiled, as you can see in this immunostaining from the OPCs. You can see individual OPCs here, corpus callosum, cortex, and hippocampus. They are all in very similar level of density. As the name says, they are progenitor cells, so they can proliferate and generate new oligodendrocyte precursors, or they can differentiate into premature oligodendrocyte, which can further mature on to forming a mature oligodendrocyte. So these are the stages which they all undergo, not only during the development, but also in the adult brain. Something which is very special about these OPCs, as it was discovered by the lab at Dwight Burgos in the year 2000, so almost like a quarter century back, was that these OPCs can make synapses like neurons. Mostly they are postsynaptic, they are always postsynaptic. And in this OPC axon synapses, they express, or they have, same kind of receptors what neuronal synapse will have, like AMPA receptors, both calcium impermeable and calcium permeable type, and NMDA receptors. And by now, there are a lot of studies which have shown that these cells also have glutamate receptors at the synapses. When these cells differentiate, they lose these synapses. So they don't use these synapses anymore. Now, with the single cell studies and also previous data from cell culture work from several laboratories indicate that OPCs can also respond to the various type of neuromodulatory signals like norepinephrine, serotonin, acetylcholine, and dopamine. And OPC, like any other cell in the brain by now, as single cell transcriptomics data indicate, also express the receptors for all kind of these neuromodulatory receptors. So the question here is that yes, OPC can integrate the synaptic activity, but probably they also very well integrate also extrasynaptic activity to regulate the fate. And the question is, how do they all put this information together to decide what it needs to and what it will be? So for this, we decided to use calcium imaging methodology in the live awake animal. We use GCAM6F, very similar sensor, which Alexey just talked about. Instead of using the viral technology as we use for neurons and astrocyte, very well, again, depicted by Alexey, we use transgenic mice. Simply, OPC cannot be transfected using AAVs. So we had to develop a very complex transgenic mouse strategy so that we can express enough level of GCAM6F in this case, and also the reporter, the structural reporter TD tomato, using the transgenic mouse line, where it is CRE-LOX-P based system, as you can see from the stopper sequence. We use NG2 CRE-ER line, which expresses CRE-ER in oligodendrocyte precursor cells, and it is tamoxifen inducible. So when we decide that when these protein, for example, cytosolic TD tomato, and when GCAM should be expressed, we give tamoxifen. We wait for 2 weeks, and then we have the cells expressing these reporters. Of course, just a quick schematic for those who are interested in genetics. I will not linger around in the genetics of these mice, that once the stopper sequence are removed, the cell will start making both TD tomato and GCAM6F. Why both? Because we wanted to track the structural changes in OPCs. Hence, to know what cell it is becoming, structural change equivalent to the fate change, which we are trying to track, and at the same time, the calcium signal when the cell is undergoing structural changes. Just very quickly, the experimental timeline: when the mice are weaned, they are 1 month old, they get tamoxifen injection to induce the expression of both the morphological sensor as well as the calcium sensor. And then we implant cranial windows, we do the training on mobile home cage, and then we do the imaging. First, these are complex genetics and newly developed mouse line. So first thing first, that does this strategy work? And indeed, it works. Most of our experiments are done in the somatosensory cortex. Again, very similar to what Alexey presented now, and in the barrel cortex. And as you can see, that there are a lot of red and yellow cells. The yellow cells indicate the cells expressing both TD tomato reporter and GCAM6F. And they are indeed OPCs because they are positive for another marker, which is PDGFR alpha. So these are double labeled cells. You can see these cells, very classical OPC-like morphology, this bean-shaped. And since once the cells are, we label the cell at OPC, and since it is a permanent labeling, when these OPCs will become oligodendrocyte, we can see them. So this is again the OPCs, some OPCs differentiating into oligodendrocyte, which we labeled with ASPA antibody. And you can see there's a big morphology change from OPC to oligodendrocyte. They become more compact cell with more circular soma. And still we have the maintenance of both the red fluorophore and the green fluorophore, our calcium sensor. So of course, the strategy works. How is the experimental strategy? We use multiphoton microscopy, and we use the glass window craniotomy. So we place a glass window over the skull of the mouse when the mice are 6 weeks old, so 2 weeks after tamoxifen induction, to make sure that we don't induce the expression of CRE in any aberrant cell types. We wait for 2 weeks. We start training the mice on mobile home cage. This is our setup. Very close up here, you can see the tail of the mouse, and mice can walk around in this carbon fiber cage, home cage. And this is the video which I took from the Neurotar. And this shows very nicely that when the mouse is trained after 1 week, a training session, 14 training session, they can be very comfortable within the cage. As you can see, the mouse is head fixed, it moves around. In some time, the mice can even fall asleep. So we train the mouse to be 1 hour happily within the cage. Later on, they are rewarded. And when the mouse is moving around, you can track it because of the magnetic trackers very precisely, and you can do calcium imaging here, for example, in neurons. How does it look like for our animals? So after placing windows, we see that we do high resolution calcium imaging. So to be able to track the morphology of the cell using TD tomato, this is an OPC here, and expressing GCAM6F. We have a parasite labeling also. So some of the parasites get labeled in the NG2 CREAR mice, which is very good for us because this helps us to go back. These are like landmark. They are very stable cells. Parasites are next to the blood vessels. You can really use them to track, go over and over again in the same area. So they are sometimes for some, it can be nuisance, but for us, they were really nice to have expression in the parasites. How does it look like? So here you will have the raw videos. Here we have a processed video. And here, when the mouse is moving in the circular track in the mobile home cage, and this is converted into speed and the calcium. So when I play the movie video, you will see that mouse is currently sitting. And since the mouse is sitting, there is not much activity going on. Once the mouse will move, you will see that there is an activity. And these are the transients. So the mouse is moving now. You see that there is increase in the speed. And also there is increase in the calcium number of the microdomains which get activated. Microdomains being this very, very small restricted area in the processes which gets activated. Mice in general, some of the mice are running across the arena, but generally they like to be very close to the walls of the arena as mice are. And you see that when this mouse take a big run, even the soma comes up. So this is what we see very consistently within the cells, that they have the spontaneous calcium transient. And when the mouse run on these tracks, they are within the cage, just locomoting around, you see increase in the calcium transient. So how do we analyze the data? So we have developed a long time back a software to analyze calcium data for astrocyte calcium transient. We called it that time Cascade. This is Cascade 2.0. We have now integrated many more functionalities in the Cascade to be able to automatically analyze both the structural as well as the calcium data. So if you're interested, you can always write me and we will be happy to share you the code. And when you look at these small area which got activated within these OPCs, this is how it looks like. So these are 92 areas in the 10 minutes of recording which got activated. You can see very beautiful calcium transient. They are faster, much faster than the astrocyte calcium transient, which you just saw from talk of Alexey. And then what we do is that we change the circular movement of the mouse within the cage into this linear track. So these are the resting phases. Here is the time scale, 30 seconds. So this mouse was for almost 2 minutes, just sitting and not doing anything. Then it tried to locomote, stop again locomotion, resting, and again the locomotion phase. So this is how you would see how we depict our mouse moving in the arena. And these are our calcium transient. So here is the same track which you saw, the mouse moving, which is the movement of the mouse in green, and the resting phase. And these are 5 domains we just chose across the cell, and we see beautiful calcium transient. They're very active, very fast, as you can see from 30 seconds. Most of them are in the range of 1 to 2 seconds. And only intermittently or very sparsely, there will be also an activation of the soma. So this is somatic calcium transient. Soma came only at the end. In general, soma did not come. One thing you see is that whenever mouse was moving, you see a lot of increase in the small little calcium transient. We show these calcium transient as this compressed heat maps. When you just expand it, it shows that yes, the calcium transients really increase with very similar timing with the locomotion. And otherwise, the cell is firing across its processes sparse calcium events. We see that resting is the phase when the mouse was sitting, or you can say quiescent or resting. And when the mouse was moving, you see that there was almost 2-fold to 3-fold increase in the activity. There is number of event, the frequency of the event also increases, but we did not see much changes for the individual event, the amplitude of the events. So mostly the frequency of the events and also the number of microdomain, these calcium microdomain increases happen within the cell. So now we have a cranial window, which we implanted. These are chronic cranial windows, so we can follow the fate of the cell. We can just track the cells, what the cells are doing over days. Some of the cells we have tracked for over a month. We track every 3 days, so we can go to the same cell and record calcium transient in the cell. This gives us quite a powerful tool at our hand. We can now see these cells either staying stable as they are for days, they proliferate, so we see the division events, and then they become premature and mature oligodendrocytes. So we can track all this. And although these events of proliferation, division in the adult brain, these experiments are done when the mice are roughly 2 to 3 months old, is rare, but we do see them. So what happened was when we see that these cells divide. So here's an example video of the two cells which just divided. So you see two cells. OPC divide in a very special way. There is just two cells just inheriting the parts of their mother cell, two daughter cells. Here is the mobility of the mouse. Red shows the cell on the left, right shows the calcium activity of the cell on the right. And you see one thing is that even when the mouse is moving, these cells, they have spontaneous calcium transients all across, or baseline calcium transient. But when the mouse moves, you don't see much happening in these cells. So just after the division, these cells were not responding to locomotion, as you see from these videos. So, but we can go to the back, because we were imaging the cells right before. So the cell looked like that before it divided into the cell like this. So one thing which we noticed is that if we call it mother cell, that in the mother cell also, when the mice were moving, as you can see the mobility, they did not have significant increase in the locomotion induced calcium transient. So the cells just before mobility, they stopped responding, or they were not responding to locomotion. And so was the case also for the daughter cells. So this is the quantification. So this is at day 0, so mother cell, and both the daughter cell put together activity. You see that activity remains similar, and there was no change in the activity between the active and the resting phase, both in the daughter cell and the mother cells. That is something which need to be noted. And the second thing which is very interesting is that activity is almost flat between the single cell and the double cell, telling that whatever receptor repertoire or whatever connectivity the daughter cell had, they inherited from the mother cell is almost like half-half. So this is the time when the daughter cell just inherited the same package of what the mother cell had, and it was just halved, let's say. Maybe a little change in the proportion, but together they are same as the mother cell. Over the time, of course, that will change. But what happens to the cell, which are becoming differentiated into premyelinating oligodendrocyte and oligodendrocyte? So this is the same cell which we imaged 0 day, 2, 6, 9, 12, and 15 days. And you see that these are the maps of the activity calcium microdomain. And this is how it looks like. So when the mouse was really active, you see a lot of calcium increase in the transient with the bouts of activity. Again, a lot of increase in the activity. But over time, you see that the baseline activity as well as the locomotion-induced activity goes really down. So this, you can already see a huge structural change, and this we know by the structure of the cell that these are premyelinating or almost myelinating oligodendrocyte. You can see here, that's what we label. And if we follow the baseline event, so the baseline activity is here at the cell, so it is going toward the reduced direction, but there is a timing after 9 and 12 days, something happens. So between 9 and 12 days, there is a huge structural change starting. That means the cell is differentiating, and you see, boom, down in activity with response to locomotion. We imaged hundreds of cells for this, over a dozen of mice. So we see this is the whole experiments put together, several months of experiment, a year of experiment put together. So we can say that there are two types of OPCs which we see. One is locomotion responsive OPCs here. So these non-responsive OPCs, sorry. So these are the LNR OPCs, which don't respond to locomotion. They have a high baseline activity, but when they locomote, they don't respond. And we saw that these are the OPCs which generally tend to divide. Then there are OPCs which are locomotion responsive. So these are the OPCs which respond to locomotion. Some of them really go crazy. And these are the OPCs which tend to go and differentiate into premyelinating oligodendrocyte 1, which is still responding, and then to more mature phase, and finally maturing into oligodendrocyte with a very low baseline activity, as well as almost no responsiveness to locomotion. So what we see here is that the cells, the OPCs, which are listening more to the ongoing activity through calcium transient, tend to differentiate. So the question rises here is that does this intracellular increase in calcium in OPC regulate the fate? Can it regulate the fate, this persistent increase in the activity which cell is seeing? So what we did is we did an experiment using chemogenetics. We used DREADs. So we use HM3DQ DREAD. This is a designer receptor, which is naturally not expressed. It's a G protein coupled receptor, which in response to designer drug like CNO will lead to increase in the calcium. And this is the pure OPC culture. And in the OPC culture, you will see that in the cultured OPC, also you have nice, beautiful transient, as we see in vivo. When we put DREAD in the OPC and applied CNO, so the DREAD expression you see with this and cherry expression, when in response to CNO, you see increases in the calcium transient, indicating that it does work in the OPCs as well. We generated a transgenic mouse line again, a triple transgenic mouse line, where we have a DREAD expression, and which is indicated by expression of M-citrin, which is a green fluorescent protein, and TD tomato, which is a red fluorescent protein. The experiment looks like this. So we take these mice, we inject tamoxifen 5 times. We want to saturate, we want to express them, our DREAD in a lot of OPCs. We wait for 2 weeks. Then we give CNO 5 days, once a day, 1 time. And during that time, we also give cells BIDU. And then we go and perform histological analysis. So then we will have cell labeled as red, which are the control cells, cell labeled as green, indicating the expression of HM3DQ, our GQ DREAD. And then we see what happens to the cell. If the cell takes BIDU, that means it recently proliferated. If the labeled cell does not label for both PDGFR alpha and ASPA, indicating that it is a premature oligodendrocyte, and if the labeled cell differentiate and become oligodendrocyte, it will become ASPA positive. So this is how we track the fate. So what happens? So these are the cells which are controlled. These are controlled animals where cells are expressing TD tomato. Thing happens here. They also got CNO. We look that most of the cell after our experiments, they were OPCs, labeling very well with PDGFR alpha. However, in contrary, the cells which had M-citrin, they did not show very good overlap. Very commonly, they were not overlapping with PDGFR alpha. However, they tend to overlap with ASPA. And in contrary, the cells which were controlled from the control animal, they were more labeled with PDGFR alpha than ASPA, quantified here. So about 60% or 50% of the cells after our 2 weeks of experiments, we see that they remain as oligodendrocyte precursor, and a very small fraction of cells here turn into oligodendrocyte, as you see here, roughly 30%. But just by enhancing the calcium activity, we doubled the proliferation rate, a differentiation rate. So double the amount of cells became into oligodendrocyte. And the cells, as you can see, also the green cells, these cells were the cells which were not more prone to proliferation, but the cells which did not hear this increased calcium were the ones which were proliferating. So just to summarize a part of the talk which I said, so we see like 2 fractions. One is the cells which are have a high resting baseline activity, and they are responsive to locomotion. We call them locomotion responsive OPCs. When the cells differentiate, they still maintain some responsiveness to activity, but over time they lose that responsiveness. There are another pool of OPCs which we call locomotion non-responsive OPCs. They don't respond to locomotion, and they tend to divide. And we saw that when we enhance the calcium, as we would predict, we will enhance the production of more oligodendrocyte. Now, the question is that where is this enhancement of calcium coming from? Remember, the mice, we showed that we found the hint that which mode of neuronal activity is regulating this OPC signal. Our hint comes from the locomotion. That what makes these OPC respond to locomotion? What is the significance of an OPC that when the mouse is locomoting around, exploring, that there is increase in calcium transient in these cells? So this comes from ours and others' past work, showing that when the mouse locomotes, when the mouse walk around in astrocyte, there is a huge increase in calcium activity. Again, very beautifully depicted by Alexey in his talk. And this is mainly mediated by the projections coming from the locus coeruleus, these noradrenergic projections. When the mouse locomotes, it's very well known, it is aroused. There's a huge bout of release of noradrenaline, and this activates astrocyte. And we thought, can that be also the case for OPCs? So what we did, we took DBH-3 mice, we crossed them to MGCAM6S so that we can now image and understand calcium transient in OPCs. And first thing we did is that we took these mice, we did the immunostaining to show that indeed the cells, these projections, which are labeled with EGFP, express GCAM, because GCAM is virtually modified GFP. And you see beautiful expression of GCAM in the processes, the cortical processes of LC neurons, co-labeled with TH, and you can even see the varicosities which these processes have. And the first question we asked are OPCs close to this projection? Are they really very close in physical proximity to be able to sense this noradrenaline if our hypothesis were true? So we did PDGFR alpha staining. As you can see, the stain of the PDGFR alpha and this EGFP stained DBH fibers. And you see that virtually some of the processes of OPCs intertwine with these fibers, indicating that they are indeed in close proximity to respond to noradrenaline release. Now we have a very good tool that if it were noradrenaline, which induces it, so there is a toxin, a neurotoxin called DSP4. If you inject DSP4 to the mice and wait for 3 days, most of the fibers are depleted or can be easily depleted. As you can see here, that when we injected DSP4, we waited for 3 days, and you see that we see more than 70% depletion in the number of fibers in the cortex. So now what we did is that we took the mouse, we implanted basically the windows here, and then we imaged the OPCs. And when we image the OPCs, you see when the mouse is moving, we have a very beautiful response to the locomotion. And then we gave mouse DSP4, we waited for 3 days, and we went to the exactly same OPCs again, because we can find them again with the help of our parasites, and you see that now the mouse stopped responding to locomotion. The data here, the control OPCs, the same OPCs here responding very beautifully to locomotion, and the same OPCs when we went back and after DSP4 injection, responding to locomotion, indicating that they indeed respond to the norepinephrine release. The question is, can OPC directly respond to noradrenaline? So we did slice physiology here. We took the slices from the same mice. We prepared the acute brain slices. We found our OPCs, and we applied phenylephrine, which is the activator of alpha-1 receptors. There are 3 types of alpha-1 receptor, A, B, and D, and it will activate all 3 alpha receptors and in the presence of TTX so that we don't disturb neurons. And we see that on application of phenylephrine, we see beautiful increase in calcium transient. Well, we done quite a bit of phenylephrine, indicating that the responses are very huge, massive. They are like almost 5, so almost 10-fold increase in the number of events. And also we see huge increase in the amplitude, indicating that yes, directly OPCs can respond to norepinephrine. The question arises, do they also have the receptors? So, well, the receptor antibodies for adrenergic receptors are not very good. So we have to do a single molecule fish analysis, more commonly known as RNA-scope. We use the probes for CSPG4 to label OPCs, which you see here. This is an OPC, this is another OPC, and these are the label for adrenergic receptor, which you see like these dots. This is for alpha-1A, this is for alpha-1B, and this is for alpha-1D. Alpha-1D is more highly expressed than other alpha receptors in the CNS. And we see that we have very good co-localization of alpha-1A, B, and D with the OPCs, which is quantified here. So all the OPCs which has more than 2 dots of these alpha-1A, B, or D, we say it expressed. And those with 0 or no dots, we say it is non-expressed. So roughly 60% of the OPCs tend to express alpha-1A, B, and D. Well, some OPCs can express probably all 3, some can express the 2, so they can be a combination of all type of expression. But at least 60% of the OPC will express one or the other type, indicating that 60% of the OPCs at least would be responsive to the noradrenaline. When OPCs differentiate into more ASPA-positive cells, which are oligodendrocyte, these receptors are significantly downregulated, almost like 10 to 15%, which also indicates that when the mouse locomotes, they stop responding when they differentiate. So now we have established that OPCs have the machinery to respond to the neuronal activity and also respond to the norepinephrine released by the alpha-1 receptor repertoires of alpha-1 receptor. The last experiment, which for today's talk is that we wanted to ask a question is that if we just stimulate the locus coeruleus by using chemogenetics, can we enhance the differentiation of OPCs? For this, we generated again a triple transgenic mouse lines where we have GCAM6S expressed. This is HM3DQ under the control of DBH, so we will activate these neurons. Experiment is such, we injected the mice, so these are not dependent tamoxifen, so hence we don't have to give tamoxifen. We just gave 5-CNO, we gave BRDU to the animals for an extended period of time, and then we tracked what happened to the OPCs. So these are the control mice which did not have expression of HM3DQ, so no staining for EGFP, and the mice expressing HM3DQ with EGFP. And then we went and quantified the number of ASPA cells, PDGFR alpha cells, and BRDU. And we saw, interestingly, is that we see roughly 15 to 20% increase, roughly, I think, 15% increase in the number of mature oligodendrocyte just by enhancing for 5 days the activity of the LC neurons, indicating that yes, the endogenously released noradrenaline has potential to push the OPCs to become a mature oligodendrocyte. And we did not see much difference in the total number of precursors, which is expected because these are homeostatic cells, and probably these cells just replace the newly generated oligodendrocytes. So again, bringing to the last part of our talk and the summary, that we show that calcium transient, the locomotion-mediated calcium transient, probably is generated in OPC through activation of alpha-1 receptors. OPCs express all 3 types of alpha-1 receptor, namely A, B, and D. When OPC differentiate, they downregulate the receptors, and enhanced calcium signaling promote the differentiation. Well, I've only presented the tip of the iceberg. There are many, many more interesting facets to the study, which I did not get a chance to present, but just to put all the things together is that we think that there are two classes of OPCs. Here you have the OPCs, which are expressing, so these are locomotion responsive OPCs. They are seemingly alpha-1 positive, and then locomotion non-responsive OPCs. The locomotion responsive OPCs tend to listen to the synapses as well as the noradrenaline signaling and tend to differentiate to become oligodendrocyte. And the locomotion non-responsive OPCs are the ones which will then replenish the pool of OPCs and or maintain the pool of themselves. And hence indicating that noradrenaline signaling, enhanced calcium signaling, can be a new mode of signaling to maybe enhance proliferation and myelination, differentiation myelination of oligodendrocytes. This study is currently in print, in press, in Nature Communications. You should be able to see it if you want to know more about it. So finally, this all work has been done by 4 of the PhD and the postdoctoral fellows in the lab. Here I would really acknowledge the work of Frederick Fiore. He really spearheaded, a PhD student in the lab, spearheaded the project and did all the experiments. The calcium imaging comes, and this analysis comes with a heavy computation capabilities, and Khalil Arase, another PhD student who did most of the analysis and generate the code for all this analysis, which we are ready to and should be happy to share. Ram and Felipe contributed to the mouse transgenics as well as the cell culture experiments. And of course, being in a very supportive environment helps to have access to the technologies, what we do around, and all our collaborators with whom we share and exchange the ideas in mouse science. Here, I would definitely want to find our funding, which is Chika and Heinz Foundation, which funds our lab, and the FOR 2289 research group from the DFG. And as you saw that most of our talks are these days very much come with a very rich graphics. So thanks to BioRender for helping us to generate these modified and treated graphics. And I would like to thank you all for your attention.
Thank you, Alexey and Amit, for those fantastic presentations. While we give you a moment to grab a drink and catch your breath before our Q&A, we have our final audience poll. And the question is, are you interested in working with awake head-fixed rats? And here the options are yes, no, I already work with awake head-fixed rats, or I would like to be contacted about Neurotar's head fixation system for rats when it is ready. So I'll just give everyone a moment to submit their responses. And in the meantime, just a reminder, if you do have a question you'd like to ask, you can submit it using the Q&A tab on the right-hand side of your screen. We may not have time to cover everything during the live session, but we'll be releasing a Q&A report within a few weeks, so please stay tuned for that. I also realize we've gone slightly over for time, and for anyone with prior commitments, I'd like to reiterate that a full recording of today's webinar will be available on demand shortly. So if you do need to leave before the Q&A, we invite you to complete the survey before you go, which I'll launch in a moment. We do read all of your responses and really appreciate your feedback. And it's now my pleasure to welcome Alexey and Amit back for the Q&A. Welcome back both. Thanks for those great presentations, and let's jump right in. So there's actually a question which relates to both of your talks regarding integration. So for example, I'll ask them both simultaneously, and perhaps you can both answer. Amit, you showed in your talk that activity in OPCs occurs mostly in the processes, and only during locomotion do you see occasional events in the soma. Is it possible that this activity in the processes integrates to generate larger somatic events? And similarly, Alexey, you showed calcium activity, integration and amplification in astrocytic soma. So, what might be the physiological relevance of this kind of integration, and may it be linked to the release of gliotransmitters? So if you could both perhaps address the question of integration in the soma, that'd be great. Maybe we can start with you, Amit.
Well, indeed, a great question. I think this is first thing which we also notice is that the activity remains in the processes. And in general, you see that most of our currently calcium imaging world comes from the neuronal world. And we see that there is always a somatic firing. And the idea is that probably there is some integration of activity going on that leads to a big increase in the calcium transient. Now, the question arises that these small microdomain-like activity, which is going on in the processes, can they come together and generate the whole cell-wide or a somatic response? Currently, knowing the physiology of the cell and knowing how they are, basically the expression profile of the voltage-gated sodium channels, as well as they are not generating any action potential, probably in general, the integration does not happen. So it is not that these small activity comes together and integrate. However, that needs to be checked more in detail, which we haven't done, but we are now doing it. That is there a threshold? If we see, let's say, 50 or 60% of these microdomain events coming up, will it really enforce now the cell-wide activity? That will be worth seeing. But one thing which we can definitely see in the OPC at least is that we cannot use now the somatic events just as a very good demarcation or a way of looking at the locomotion activity, which we could do for neurons. So for neurons, whatever happens, you can see in the soma. In OPCs, definitely not. There will be a lot of things which are happening in the processes.
And Alexey, about the astrocytes, similar question.
Well, it has to be studied further, but I can speculate based on the knowledge about astrocyte morphology and organization of the cells. So astrocytes have soma and branches which are filled with organelles, with mitochondria and the plasmic reticulum. They also have fine leaflets which fill the space between synapses. And there is a lot of calcium activity which occurs in this fine leaflets around synapses during synaptic transmission. So we have NCX, sodium calcium exchanger, which triggers local calcium response. And this local calcium activity can be amplified also by release of calcium from endoplasmic reticulum. And probably that's what modulates synaptic transmission. And that's because it's just colocalized with the synapses. And so probably if any release of glial transmitter occurs, it might be occurring in response to these local calcium elevations. So then if calcium activity propagates into soma, I don't think there are so many synapses surrounding soma. Of course, there might be some, but amplification of calcium response in soma maybe is a signal for mitochondria, because regulation of metabolic processes by calcium has also been described. And I would say distal calcium, the calcium which occurs in the periphery or with in areas close to synapses, would be calcium which regulates synaptic transmission or has some other signaling role or release of glial transmitters. Calcium which amplified in soma probably has metabolic sort of message. So it's kind of a metabolic signal and regulating maybe growth of astrocytes or shrinkage of astrocytes or something like that.
Okay, wonderful answers. Very interesting. Detection of this, we have a related question here. I think detection of this is obviously contingent on high resolution images. And if you're studying that under locomotion, obviously the stability of the image is a challenge. So another question for both of you would be, are there any additional steps required for stabilizing of the images that you're acquiring? Maybe if you can expand on that a bit.
So I can take on that, Andrew,
Sure.
and if it's okay for Alexey, is that, yes, what we see is that in general, the recordings are quite stable, even when the mouse are well trained to be under the mobile home cage. They need to be trained. I want to reiterate that, so that the proper trained mouse who has really learned to be under the mobile home cage, we do a training for a week, is very, very calm. Don't try to move the head too much. And the movements are also very flat and not erratic. And that leads definitely to less mobility. So when the mouse is moving, especially when it starts to move. And the second thing which we found is that since we were doing the double imaging, one is the morphology imaging by using TD tomato, and the other calcium sensor, which is GCAM, it also helped us because now we use it like a pseudo ratiometric analysis, because what we can do is that we can always divide our calcium traces with the motion traces, if you will say, any changes in the fluorescent of the TD tomato. And that really not only helps us to weed out those little jitteriness in the data, but also we see that quite often you will see this mobility artifact, which might sometimes match like a calcium artifact. So here, a very careful training of the mouse have to be gentle and less stressed, and TD tomato does help.
Sure. That's a great answer. Alexey, anything to add?
Sure. Firstly, I would agree with Amit about stability of the system. So mobile home cage is very stable, and we don't generally have serious problems with image movement or instability. However, in our pipeline of image analysis, we have a step for correction of the movement in XY direction. So that can be done post hoc during the analysis step. But if there is a movement in a Z direction, so that would be a problem if we do calcium imaging. Again, we don't have much of Z movement, but if it occasionally happens, so we have to discard this data, because then, of course, we can have artifacts related to the movement of the sample, and that could be detected as calcium activity by the software. Obviously, that's a general problem. Everyone who does calcium imaging in vivo knows about these things. And if it comes to morphology, so then it's easier because you can do Z-stack of the cell, and then there is no problem in Z movement because you can correct this also during your analysis. So I think it's a problem, but not that big problem which would make our experiments impossible.
Okay, wonderful answers. Maybe we'll another question here. We'll continue with you, Alexey. You mentioned the refractoriness of the calcium response in astrocytes, which is really interesting, and maybe makes them quite distinct from neurons. Could you speculate on a possible mechanism for this refractoriness?
Sure. Calcium responses in neurons and astrocytes, they have very different mechanisms. In neurons, probably bulk of calcium coming from voltage-gated calcium channels. So during each episode of locomotions, neurons fire action potentials and trigger voltage-gated calcium channels, so there would be a calcium entry through plasma membrane. In astrocytes, calcium response is much more complex, and if we're talking about this amplified response in somas of astrocyte, most likely it depends on the calcium release from endogenous calcium stores. And IP3 level, generation of IP3, and also amount of calcium in calcium stores. So, and depletion of calcium stores during the first episode of locomotion. Second, inactivation of IP3 receptors or G protein coupled receptors, which trigger this response, could all contribute to this refractoriness. And so it has to be studied. We have to look at it. But there are some reports from the lab of Hajime Hirase showing that if you do genetic stimulation, which triggers release of noradrenaline in each of the stimulations, so your calcium response will get smaller and smaller. So it's possible that there is an activation of receptors, but of course, it might be much more complex phenomenon than that. But this refractoriness itself is quite interesting because people thought that calcium response in astrocytes is important for regulation of local blood flow and some regulation of the blood vessel, vasomotion, etc. And probably this refractoriness is inconsistent with this hypothesis, because that would mean that during one episode of locomotion you will have vasomotion, during the second episode of vasomotion you will not have. So that's I think has important physiological implications.
Yeah, that's fascinating. Really interesting stuff. We've run a little bit over time here, so maybe we'll just finish up with one final question. This one for Amit. Given that the norepinephrine signaling is promiscuous, how would such neuromodulatory signaling exert specific effects on cell fate regulation and myelination?
Oh, that's a hard one, I must say. A really interesting question related to, yes, how would noradrenaline signaling regulate the fate? Well, we can, again, here speculate. We have some ideas from our experiments. And it's the question which we are really following also, is that how it does what it does. So there are some clues which comes from the expression data which we have, and also what we see from the single-cell transcriptomics data, that these cells, so now you can really expand the lineage of the cell. You can really look at the different part of the lineages of the cell. OPCs, pre-myelinating cells, they also come in different names. You can also call them committed oligodendrocyte precursor, mature oligodendrocyte also comes in different flavors. And you see that there is certain stage of OPCs, they tend to express the IP3 receptor expression. as well as the receptors for the G protein coupled receptors, like alpha-1A and alpha-1B. Usually alpha-1B comes up quite high. And the idea here might be is that the cells, probably they are now synaptically connected. They're listening to the neurons. They're getting their own share of calcium by the regular neurotransmitters like glutamate and GABA. They changed to, they said, okay, great, something is going on. There is a moving, the fate decision has been made that, okay, great, let's move on there. Now they start to upregulate with this fate decision, let's say IP3 receptors and adrenergic receptors. And hence now they become responsive to the activity, which they take it through towards the completion. So the idea is that there can be two steps. First step is that they are now making the fate decision, which probably is not dependent on noradrenaline, which is done by, let's say, the synapses and the glutamate receptor signaling. And once that is done, the noradrenaline signaling, the ones which can sense it, can really carry over. One which does not sense will die out and will not be there. And it can have really very interesting implications. For example, in multiple sclerosis, we have in the lesions a lot of stalled OPCs. And can we now use this mode of signaling to really push? And there are a lot of antidepressants being promiscuous. The good thing is that there are a lot of antidepressants which work on preventing either the reuptake of noradrenaline specifically, or even the noradrenaline and serotonin. And already MS patient takes antidepressants. So basically this can be probably, maybe if it works, can be just change of certain medications and to promote myelination in these patients. So these are the experiments which we are currently trying to do, is to push this signaling towards remyelination.
Well, it's fascinating stuff. I don't think either of you have a shortage of interesting directions to move into. It's fascinating work. Unfortunately, we are out of time, a little bit over, in fact, but I'd really like to thank you, Alexey and Amit, for sharing all of your insights with us today and the fantastic presentations. It's been an absolute pleasure having you with us.
Thank you for the invitation.
All right, thanks very much, both. A big thank you also to everyone in the audience for participating and staying with us today. And last but not least, we'd like to thank Neurotar for making this event possible. If you haven't already, we now invite you to take a moment to share your feedback on the survey, which you'll be directed to in a moment. We do read all of your comments and greatly appreciate your input. And if you still have a question, it's not too late. Feel free to submit it via the survey, and we'll make sure to forward it along to our speakers following the event. If you're interested in learning more about the research presented today, we also invite you to check out the resources panel. And in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Thanks, everyone.